Good morning, everyone, and welcome to this IHI Impact event on blood cancers, and more specifically, how the Innovative Health Initiative projects are leveraging big data and CAR-T therapies to improve outcomes for patients. My name is Oussama Karoum, and I'm Scientific Officer at IHI. So before starting, just two quick points. The first, that the session is being recorded and that the recording will be made available on IHI website and in our YouTube channel as of this afternoon today. Second point, you can use the chat box, which is at the bottom of the screen at any time during the webinar if you want to ask questions to our distinguished speakers. So first of all, I wanted to give you some key figures about cancer in the European Union so that we can kind of set the scene. So more than 2.7 million new cases, new persons are diagnosed with cancer every year in Europe. So what does that mean? It means that in practice, 1 in 4 women and 1 in 3 men are expected to be diagnosed with cancer before the age of 75 in the European Union. And in terms of mortality, 1.3 million persons die from cancer every year, again in Europe. And again, in practice, it means that 1 in 11 women and 1 in 7 men are expected to die from cancer before the age of 75 in the EU. So this gives us this shows us how the cancer is a burden for European society and how it's a high unmet public health need. When you look at the figures for blood cancer, specifically in the EU, we see that more than 200,000 patients are diagnosed with cancer every year in the EU, and that more than 100,000 patients die from blood cancer every year in Europe. So for all these reasons, IHI has funded and launched many projects in the cancer field, covering 14 different cancer indications, going from the most common type of cancer, such as lung, breast, prostate, and colorectal, as you can see on this screen with the title of the projects, but also more rare cancers, such as sarcoma, brain, or ovarian cancers, for instance. But today's webinar will be focused on blood cancer, where you can see that we have several projects which are focused in this indication. So as you may be aware, September is the Blood Cancer Awareness Month to raise awareness about blood cancer and for example, like leukemia, lymphoma, and myeloma, which are the most common type of blood cancers. And we have decided at IHI to try to extend this awareness month by 1 single day until today, the 1st of October, to raise awareness about this, about blood cancers. To do so, today we will have the presentations of 4 different projects. So 2 projects, namely Harmony and Harmony Plus, which have been working on how big data can improve the outcomes for patients with blood cancer, and also 2 other projects which are focused on supporting and advancing the development of CAR-T cell therapies, these projects being namely T2Evolve and EASYGEN. So let's start with the first project, which is Harmony, and to do so, I have the pleasure to welcome 2 distinguished speakers, which are Jesus Maria Hernandez. Jesus is a medical doctor, hematologist, and professor at the University of Salamanca in Spain, and is also the Harmony and Harmony Plus project coordinator. We also have the pleasure to welcome Lars Bollinger. He's also a hematologist and a professor at the Charité University Berlin. And he was also involved in both projects, Harmony and Harmony Plus, as a work package coordinator. Jesus and Lars, the floor is yours.
Well, hello, good morning, everyone. First of all, I want to thank Oussama and IHI for the kind invitation to this meeting. That this is for us is something really very special. And I'm also very happy to see the previous slide in which we have seen a lot of oncology projects in the framework of the IHI, and we were really one of the first one in oncology. And this is something that is really providing us a lot of, let's say, really very nice feeling about this. How is the Harmony Alliance? The Harmony Alliance is composed by Harmony and Harmony Plus projects. Both of them were funded by IHI, Harmony, in fact, by IMI. And we are mainly focused in hematological malignancies. So we are focusing cancer in blood cancer. And this is we established a really network in which we were able to put on the same table all the stakeholders active on the field, starting from the patient associations involving bioinformaticians, involving regulatory agencies, and involving also industry, that this is always 50% of these projects, but also they were really mainly mostly involved in many fields of the project, together with the academic, together with hematology, networking associations, and so on. We'll move on to the next. Okay, in these two projects, we have built a community of approximately 500 professionals working on the field. We are receiving data coming from 140 data providers from 34 countries, most of them in Europe, but also out of Europe, mainly in Harmony Plus project. We worked with the 9 European patient organizations, with 9 pharmaceutical companies, and we identify over 160,000 cases, and we were able to harmonize to OMOP. This is the common standard to do a harmonization over 100 databases over the world. In that way, in this program, in Harmony Alliance, we were able to build a robust big data platform involved, and now we are running and finishing over 30 collaborative projects. Next slide, please. What are the results? So first of all, it is the alliance. The alliance is built by these over 100 organizations over the world. Now, maybe one of the organizations in which you are working is already there, and I hope we didn't forget one. That this is quite normal. That forgetting in that point. Next one. Next. So this is the alliance, and this is the numbers that we get from the different countries. Harmony started in the European community. It's an IHI, so it's Europe project now coming to the other places over the world. But this is the composition, or this is mostly the data barometer that we had performed until this year. And these are the data coming from the different countries, mostly Germany, Netherlands, UK, Spain, Finland, and also Eastern countries as Czech Republic or Poland. Next one. This is the data cycle, and the data cycle sometimes is quite difficult to be understand, but it is important to know that Harmony started before the GDPR was put in place. So we have to do in our data lake, all the data are anonymized data in a way in which we are performing a double-sided anonymization with some guarantees and working with a trust third party in the middle. So this is providing many data access restrictions. So all the work, all the projects are running inside the database, and nobody can take the data in his computer. And in that way, we are working with anonymous data, and this is there are many I will say, organizational measures to assure the data and the data restriction to the database. So all the data are anonymized, all the data are managed in an aggregated way. In that way, it is we are compliant with the GDPR and the local registration. Next. And this is the data journey that we want to, I will say, it's quite difficult trying to explain this, but overall, we identify the data sources, we perform this anonymization that I explained, and we map all this data to OMOP. OMOP is a common standard that now is used also in other projects, in other IHI projects, and in other European projects as Darwin, Eden, and so on. After the harmonization is, no, okay, this is important to do a data curation because it is sometimes the harmonization is providing some problems with the data, so it's important to do a curation with experts and with a scientist, data scientists working on the field. And we have established also some visualization tools in order to better analyze which are the data that we are capturing. Later, we provide the possibility to do analytics inside the project, and the data are going to be disseminated and published and communicated in the best congress. Which next one, please? We go, okay. So in that way, we have developed some tools, and data harmony was based on patients. Most of us are hematologists working directly with patients, and we really want to do or to elaborate to do some tools that could be used at practical level. So we have some data exploration tools that are available in the Harmony platform, in the web page, in the Harmony Alliance Foundation page. And also we have established a couple of outcome predictors in AML and multiple myeloma. So these tools are free to be explored by hematologists and to really calculate the risk of AML, the risk of multiple myeloma by using this tool from any kind of device in front of the patient. And all these tools are based in this high number of patients that we have achieved, over 30,000 of AML and over 15,000 multiple myeloma cases. Next one. Maybe Lars can explain this AML tool, that is the data visualization, as an expert in AML, and he was built this tool. Lars, please go ahead.
Yeah, good morning also from my side, and thanks also for the invitation to this meeting, and of course also to the IHI for supporting our projects. Yeah, I'm a hematologist, oncologist who works mainly with acute leukemias, and the pleasure for me was to organize the work package that was identifying the data sets and putting them together, and also, let's say, coordinating the research projects. And one of the project's results you can see here for acute myeloid leukemia and acute large lymphocytic leukemia, like also for the other hematologic diseases which we have put together in Harmony. And the plan was here to provide a comprehensive picture of hematologic malignancies in Europe. We wanted to, of course, also develop a tool that allows people who want to use Harmony to get let's say an easy overview of what data is captured in Harmony, and what information is available that could be useful for your research questions. And with this tool, you're happy to look at the tools through the Harmony platform. You could have seen the link on the previous slide. You can explore, for example, the AML data set in a way that you look for specific gene mutations or cytogenetic abnormalities you are interested, or for example, for also gender aspects, age-related aspects. And the tool allows you to get a first glimpse. And then when you think that the Harmony platform could be useful for your research, then as I will show on later slides, you are also then available to address your research question. And then you can get access to the data and work with the platform. Next slide. Yeah, this is what I've already explained. So it's an open platform. We have many ongoing projects in AML. This is now still 10 projects where we follow up on things which we have started during the active funding period in Harmony. And of course, we also want to keep the platform alive. We are currently also embarking on continuing to collect new data sets, put them in Harmony, where you then can use them for your additional or new research questions. But in the long run, of course, we think that you can then also use Harmony to deposit your data in a safe and secure way, to also fulfill the need to make data publicly available to the research community. So all of you are, of course, welcome to get in touch with us. And then on the next slides, before I then hand over again to Jesus for the impact of the entire project, we have brought to you also some more concrete research, let's say, results from Harmony. For the AML project, we have developed tools that allow you to predict a patient's individual risk based on the underlying genomic abnormalities and the type of disease, that can help you predict outcome to conventional chemotherapy or allogeneic stem cell transplantation. And what we do here is you can type in your patient characteristics on the left side in that tool, including the cytogenetic and molecular genetic abnormalities. And then the program, using also machine learning algorithms, compares your patient's individual profile to a respective cohort in the Harmony platform, and then plots the patient's risk with regard to overall survival, relapse-free survival, as exemplified here. But you, of course, can also look for other parameters, for example, response to therapy or the chance of becoming MRD negative. And then based on your individual patient's profile or the group that has the characteristics of your patient in the Harmony platform, which would be the blue curve here, you can easily see that the patient is doing as well as a favorable risk cohort according to your European Leukemia Net guidelines. And based on that, for example, it's easier for you to make the decision that that patient might have a high chance of being cured with conventional chemotherapy and that you would not have to move forward to allogeneic transplantation. On the next slide, you see a respective example for multiple myeloma, where we also have put together a large data set, and this also has been published. Here you, for example, can mine your patient's laboratory parameters and see what his outcome risk would be. And based on that, you can then determine whether your patient has a high risk of overall survival and progression-free survival. So those tools are not only useful, of course, for your daily clinical management, but the data underlying these tools, as I had said, can be mined for novel research questions in the future, and we will continue to enlarge the data sets to, for example, for AML, also reflect now the novel targeted molecular therapies. And with this, I think on the next slide, I will hand back to Jesus. Oh no, this is just one more example of the publications that have come out from Harmony. Projects on AML have been published. We had put together a large data set on acute promyelocytic leukemia under the lead of Maria Teresa Boso. There is data from MDS that has been published, and we didn't show all the other publications that came out on myeloma and CLL. So the nice thing is also in Harmony that the different countries, different PIs we're taking the lead based on the research questions they brought into the platform. With this, we're also quite flexible, and of course, you're also welcome to lead your own research projects in Harmony. And on the next slide, I think now, I hand back to Jesus for the impact of the entire project.
So it's just to maybe you can see easily that we were able to cover not all hematological malignancies, but most of them, and the more important one, we were working in AML, multiple myeloma, acute lymphoid leukemia, chronic myeloid leukemia. This tool that Lars already explained about multiple myeloma is going to be published in Hemasphere this month. It's not yet published, but it is going to be published this month. So, and all the tools, because it was some question on the chat, are available directly from the web page. So, together with this impact, in order to help the clinician, in order to provide some scientific good quality papers, we have been invited to be a member of Darwin network. Darwin is a very huge, important project from the EMA, and we are really proud to be there as a alliance, as a foundation, not covering several institutions in our framework. And this is really a great pleasure to be there, and also a good opportunity to continue doing the best mapping as possible together with all these really very, very important in big hospitals across Europe. Next slide, please. So, which is we think we are providing, and we are going to provide more and more value for the patients, and we have to work together with them in order to incorporate the quality of life or life lab data, the pros and cons from them. We are providing, and we are going to provide more and more data, helping data for good quality data, to help clinicians to guide them in the clinical decision at the clinical level. Next one, please. Next slide. And also, we think that we are going to provide also, or we are providing pharma value for the pharmaceutical companies, because we are generated synthetic arms for single arm, and we can do external comparison with a very high number of patients. And Darwin is a good example of this. It is something that also we made possible to do a value for a regulator and HTAs agencies. So in that way, we think we cover any kind of thing that we were thinking from the very beginning in the project. And now, next one, please. Now, for all the stakeholders working, so what we are asking is for receiving research proposal, and also to continue to work by providing data, by providing feedback, and by using the data that Lars already explained to all of you. Next. So we think that Harmony is working in not coordinating hematological community. Hematological community is a very large community, but we try, and we were doing this job in the last 8-10 years, and we continue to work on this field, taking in the same, sitting in the same table, all the stakeholders working on the field. And this is one of I think this is important, and these are the advices that we receive from IHI, and it is not easy, but it is something that we are more and more doing by incorporating patient organizations, by incorporating HTAs, regulatory agencies, and also academic. Next one. And now we have the next step of the both projects was the creation of the Harmony Alliance Foundation. Harmony Alliance Foundation is working in the last year, so we are now a little bit more than 1 year old, and our mission is to. Sorry, could you come back one? Our mission in Harmony Alliance is to enable data, to fostering collaboration, and in order to transforming patient care in hematological malignancy. That this is really important. And the vision is to empower the hematological community to share and harness data, advancing research, and driving innovation to improve the lives of patients worldwide. So we started with data. We started with good quality data. We incorporate OMIC data. As you have already seen, we have data coming from genetic lesions in the different hematological malignancies. And now, after the incorporation of all the follow-up of the patient response to the therapy, survival, relapse-free survival, we are able to really provide a very important, very comprehensive information about this patient. Maybe we can get the last one. The last one. So for this reason, we invite to all of you. We really want to thank one more time to IHI for all the support that we have received from them. It is quite important to be together, to continue together. In our mind, the projects must end in one way, in one day, but also we have to continue the collaboration, the cooperation with all the hematological stakeholders, and also with the IHI office that provide the umbrella to do this. And this is really very important for all of us, and we are really happy to receive any kind of questions. Thank you so much for being there, and this is really very important for all of us. Thank you so much for the invitation, and happy to take questions.
Thank you very much, Jesus and Lars, for these very good presentations. It's an impressive work. Now Harmony project, both Harmony and Harmony Plus, have ended. And we've seen the enormous amount of data that you have managed to succeed in collecting in your centralized big database that is now open for researchers to submit research questions, which is really key, I think. And also, it's very important to see that you also developed some very useful tools for also physicians, but also patients, and that you will continue within Harmony Alliance this work, growing the big data platform and developing further those tools. So thank you very much. We already have some questions coming, but these questions will be answered at the end of the webinar during the Q&A session. So now we are moving on to the next project presentation, which is T2Evolve. And for doing so, I have the pleasure to welcome Michael Hudeszek. Michael is a medical doctor and professor at the University Hospital Wolfsburg in Germany. And Michael is also the T2Evolve project coordinator. Michael, over to you.
Thank you for the very kind introduction and the invitation. I hope you can hear me well. T2Evolve is a project then that is also has a focus on hematology, but also takes us into the space of oncology and also non-malignant diseases. And the topic that we're covering is ATMPs, so advanced cellular therapies with gene-engineered immune cells, CAR and TCR-modified immune cells to treat these diseases. For those that are not familiar with this topic, so over the past 10 years, there has been a tremendous development. And there's a few very also prominent patients that have experienced the potency of CAR T-cell therapy, including Emily Whitehead, one of the first pediatric patients, now more than 10 years ago treated with this therapeutic intervention for acute lymphoblastic leukemia and still in remission, illustrating that this is a transformative treatment to which we want to give access to many more of our patients than we currently can do. And this is why we've embarked on this project in T2Evolve. The therapeutic principle is that we take immune cells from a patient and we gene engineer them to express synthetic immune receptors that can recognize surface molecules on tumor cells and re-administer these immune cells as an infusion and an autologous immune cell therapy. The next slide. This therapy is also on the market, and there's approval for several commercial CAR T-cell products in the space of hematology to treat leukemia, lymphoma, and multiple myeloma. And there's a surging global development to bring this therapy also to other indications in hematology, for example, treating myeloid malignancies, but also in oncology to treat the actually much more prevalent solid tumors like breast cancer, lung cancer, prostate cancer, etc. And the motivation to bring this consortium together was certainly grounded in the observation that even though there is a lot of innovation taking place in Europe, when we look at the access of our patients to this novel treatment modality, first in the form of clinical trials, later on also then with approved immune cell products, we see that there's a kind of a gap between the activity that is happening in the United States and in Asia, mainly in China, and in Europe. So this is the clinical trials world map from 2021 when the project started, where we see that Europe has activity with at that time a little more than 60 clinical trials, but this is shy of the activity that is happening in the US and other places. And the next slide. Even though there is so much creativity, so much innovation taking place at European research institutions, we considered that one bottleneck is the speed and also the determination with which we can bring these innovations then into clinical trials. This is why we use these road signs to illustrate this bottleneck and what we're trying to accomplish in T2Evolve, and this is to actually create a bench to bedside and bed learning cycle and accelerated learning cycle, where we can bring these innovative immune cell therapies into clinical trials, do our correlative studies, and take the insights and learnings back to the drawing board and into the lab to develop the next iteration cycle and the next wave of these immune cell therapies. And the key foundation in order to accomplish this is that there are standards in the preclinical development in terms of what models do we use to determine preclinical safety and toxicology, but also efficacy of these immune cell products, but also what are standards for the manufacturing and release of these cell products under good manufacturing practice, and then the clinical use in first in human or later phase clinical trials, and the analytical monitoring and correlative analyses that we do in order to learn as much as possible within each trial, but also between trials in order to improve our ability to learn between trials that, for example, target the same disease, but with different cell products. And this has defined then also the working pillars in T2Evolve that was composed in 8 work areas and work packages. Let's go to the next slide. And reflected by the expertise of the partners. So the vision that we created at the time that we wrote the grant is that in the future, at the time, and the future is now, scientists come together with a product idea, so an idea for an innovative immune cell therapy, are able to generate the preclinical data in models that we know are accepted by regulators and are informative and predictive and can go into the clinical trial application in order to accomplish the approval for clinical trial rapidly, and then be able to implement these clinical investigations at European hospitals such that patients in Europe get access to these innovative therapies. Working with not only preclinical models, but also standards, as I said, for manufacturing and release, for correlative analysis and diagnostics in order to strengthen the innovation ecosystem in Europe. Now we are almost towards the end of the funding period for T2Evolve, and I can tell you that we have made substantial progress in accomplishing this mission. And let's move on to the next slide and to provide you with a few examples. Let me first, however, show you the partners that have engaged in accomplishing this mission. So it's more than 30 partners, academic institutions with expertise in CAR, but also TCR, T-cell receptor engineering, biotech companies like the biotech spin-outs from some of the academic partners, including T-Curix, for example. There are major pharmaceutical companies and stakeholders, but also policymakers and patient representatives and regulators involved as the partners in the consortium. And also we've been able to create a very active stakeholder community. Very soon after initiating the project, we've been contacted by many entities, European, but also beyond Europe, asking can they become part of the consortium. And in order to keep the consortium operational and able to work, we've created the stakeholder community. We can go to next slide. Where we created different formats of workshops, public meetings, such that also entities that are not consortium members, but that are active in the field of cell and gene therapy can participate and benefit from the progress and developments that we made in the project. We've been trying to be very creative also with the formats that we tested and used for this interaction. Of course, classic roundtable discussions at scientific and medical meetings, but also panel discussions, patient and family days, so-called science cafes that were dedicated to specific topics of interest in this field and that engaged multi-stakeholder and multi-expert individuals, but also workshop events that we hosted often in conjunction with national or European scientific and medical meetings. And this has allowed us to really create a consortium and an ecosystem that is very active, that is also quite visible in the field of cell and gene therapy in Europe, but also beyond. Let's move on. Yeah, these are the work areas that we focused on. There is, of course, a strong interest in preclinical models, and we, of course, tried to reduce the use of animals in preclinical safety and toxicology, but also in efficacy testing, but also paid particular attention to working with methods and models that reflect the tumor diseases that we actually treat in patients, and have documented predictive value. And we've been quite active also in engaging with the European Medicines Agency Innovation Task Force, for example, in determining what innovative models could be integrated into IND applications in the future that allow us to obtain these data more rapidly and obtain also an IND approval more rapidly. Analytical methods, harmonized registries, and regulatory framework also play into this topic of the desire to accelerate the clinical translation of these innovative immune cell therapies. And a very important aspect, and this has been also one of the, I would say, most enjoyable parts of the project, is really the interest and engagement from patients that have been very active. And we have a dedicated patient group, also patient advocates with different disease backgrounds, I would say, and they've been very active and engaged in the project. And this has allowed us to also make substantial progress in our efforts to improve the access of patients to information and education materials, but also to map the access that patients have in different areas of Europe to these treatments and ways to improve this. I'll give a few more details on some of these areas. Let's go to the next slide. This includes a survey that we've conducted, and this is to date the largest survey that has been conducted amongst patients that have actually received these innovative immune cell therapies, CAR T cells, and the paper has just been published a few days ago, to really understand how was this CAR T cell therapy from a patient perspective? Are there things that we can improve in the informed consent procedure? Are there things that we can do better to engage also the healthcare providers? And this is not only the medical professionals, but also, for example, patient families and friends for the short-term, but also the long-term follow-up that is necessary after this treatment. And we've been able to translate the survey into multiple European languages in order to increase our coverage. And this is also an activity that we're intending to now continue and perpetuate, I shall say, to be as inclusive as possible and also make the survey available to more patients in additional geographical regions in Europe. Let's move forward. One of the key objectives of the project was to facilitate the clinical translation and therewith the evaluation of innovative CAR-T and TCR-T therapies in first-in-human clinical trials. And even though none of these clinical trials has been directly funded by the project, the mission of T2Evolve was to facilitate the clinical translation by creating these standards, such that the work by the IND sponsors was more feasible, faster, and also could be performed with the means and infrastructure that is available to them. And therewith, I can say that we've been able during the project lifetime to witness the initiation of multiple clinical trials. They come from different academic partners in the consortium and include many first-in-human clinical trials that also are landmarks on a global perspective in terms of evaluating novel targets and bringing CAR T-cell therapy and TCR-T therapy to novel indications that has not been tested at other institutions worldwide. Let's go to the next slide. This is also true for the biotech and industry partners. Also, the biotech partners have been able to launch now first-in-human clinical trials from their own pipeline, but facilitated by the models and standards that have been established in T2Evolve. This includes an AML, acute myeloid leukemia CAR T-cell trial by TCURX. And also the industry stakeholders have increased their commitment to Europe and their activity in Europe and fostered the development of their CAR T-cell products targeting BCMA and multiple myeloma and CD19 in leukemia lymphoma, but also in autoimmune diseases in Europe and at European sites. So I think overall we've been able to facilitate and make, let's go to next slide, a substantial contribution to in our desire and aspiration to really bring more of these therapies closer to our patients and increase patient access. And if you all of this in a space like ATMP therapy cannot be done without considering the regulatory framework in which we operate. And therefore, we've been able to establish and maintain a very productive and constructive dialogue with EMA, but also with several national regulators, and also reflect on some mechanisms that could be used when interpreting the current legislation, but also when shaping the evolution of this legislation in order to make it compatible with the rapid innovation cycles that are currently ongoing in ATMP development in the research laboratory. One example is the so-called parent-child concept, meaning that by the time that we bring an immune cell therapy to the clinic, we've already developed an improved version of this in the laboratory where we know it's potentially more effective, it's potentially safer, and we have the desire to then bring it into the same clinical trial that we're already doing, or bring it into a follow-on clinical trial rapidly. And this is what can be facilitated by the parent-child concept, which is catered for the regulation in the United States. And we've worked hard with EMA and the regulatory experts in the consortium and published a reflection paper on how this concept can be adapted to the European legislation. And we've identified several projects now in the T2Evolve consortium that are using these regulatory insights and advances in bringing more innovations to patients faster in Europe. There's a number of policy briefs that we've published and also reflected on the access of patients to CAR T therapy. Of course, there's this is important to consider also the ability of the national healthcare systems and the ability to reimburse these therapies that are not cheap. And therefore, we've been able to map together with our patients and the experts that we have in different European member states, what is the de facto access of patients to this treatment and how can we improve this access either by working in the respective countries or by also creating the ability to refer patients to sites that have access to this therapy. Let's go on to the next slide. A few more examples. So we've been able to really work on, establish, and also publish on standards in all aspects of the CAR-T and TCR-T development cycle, preclinical, manufacturing, clinical use, and there's a number of papers that have been published in very visible journals in order to share these insights with the community and also make it accessible to our stakeholder community, but also to other stakeholders in Europe and worldwide that are interested and are active in this field. And we have very close interactions also with CAR-T experts in other geographical regions worldwide. Examples include the BioCanRx network in Canada. We just had last week colleagues from Australia and New Zealand visiting our site in Würzburg, and we were able to share many of our insights and offered these colleagues to adopt the standards that we've established in order to facilitate the implementation of CAR T-cell therapy programs at these sites. So this is really a global activity that we've established here, and we're proud that Europe is the center of this activity. Regulatory engagement is important. As I said, also patient and healthcare professional engagement. I think we've been quite active and also been able to reach out and relate to other European projects, including projects in the IHI initiative. And as I said, we have a very active community of stakeholders in this field of cell and gene therapy. The next slide. A few metrics in order to also, try to make measurable of what we've accomplished. So together with the experts in the consortium and national and EMA regulators, we've discussed more than 5 innovative models that are either faster, better to implement, better scalable, animal-free, or more predictive in the preclinical assessment of CAR-T and TCR therapies. 3 of them have already been adopted and are currently used in ongoing IND applications. An additional 2 models are currently under evaluation with our regulatory experts. We've been able to identify several new manufacturing sites, both at the academic partners, but also at the biotech and industry partners that are now active and operational in Europe, such that there's more CAR-T manufacturing actually happening in Europe in order to make the production easier and also create the opportunity for valorization in Europe and not in other areas of the world. As I showed you, we've brought more than 9 novel CAR-T products into clinical trials that are already active and recruiting. We've been very active at scientific conferences, meetings, on the social media, and have a strong group of followers, also on LinkedIn, for example. There have been numerous publications. We expect to have more than 30 papers out by the end of the official funding period, and there will be more publications to come. And numerous standardized processes in the form of validated SOPs have been tested and implemented at multiple sites, both academic and public, in the consortium. So moving forward. This is what the CAR-T clinical trial roadmap looks in 2025. So I think there's a substantial improvement in Europe. So we've gone up from approximately 60 trials to almost 200 within these 5 years. This is not, of course, solely the accomplishment of T2Evolve, but I think we've created a framework and a think tank for this therapy in Europe that is very visible and has been accessed and used by many stakeholders. We also see that, of course, the US and China have also increased their the rate or the number of their clinical trials, but still I think we're on a good track and show that we can make it happen in Europe. And therefore our desire is to also perpetuate the work and find a way to continue and still expand the ecosystem that we've created beyond the EU funding period. A few more just metrics and then considerations. I think we've been able to expand the number of clinical sites with expertise in CAR T therapy quite substantially, also through our interaction with scientific and medical meetings, including, for example, the European CAR T-cell meeting, which is to date the largest scientific meeting dedicated to CAR T-cell therapy. I think we've been able to map and identify ways of making sure that there's equitable access of patients across Europe to this novel treatment, and also consider of how this treatment can be made more affordable and also socioeconomically more sustainable. Point of care manufacturing, the implementation of GMP facilities is part of this effort, because the ability to produce the sub-product also close to the hospital is a key element of this. And again, this requires then standardized models and standards in the preclinical development to show the manufacturing process is robust and safe, and the manufacturing itself in order to then provide the treatment to the patient. Next slide. So in looking ahead and looking forward, we have kind of condensed the spectrum that we are currently working on into kind of 4 key work areas that we also want to now continue in form of an association that we're currently incorporating and launching in order to have a seamless transition for T2Evolve from the official EU funding period to now an association that is still able to work and continue the work that we've started. For this, we've identified the area of patient engagement and education as being particularly interesting and valuable. Of course, regulatory innovation and the identification of roadblocks to patient access. Frontiers in innovation, so the continued investigation. No, let's go back to the slide. Let's go back. The frontiers in innovation with innovative preclinical models, standards, and also manufacturing concepts, all the way to in vivo gene transfer, which is already on the horizon. But then also the area of clinical translation and correlative research. These are the 4 key pillars that we've identified and that we want to continue. Let's move forward. And since we've, let's move forward 1 slide. And as I said, in December 25, the official funding period will end. The consortium, though, is highly enthusiastic and wants to continue. And we've identified several stakeholders, both academic, public, but also from the industry that is willing to support the association. This is why in the month of October and November, we will incorporate the T2Evolve Association such that we can continue the work that we've started. Let's go 1 slide forward. This is what the management team has created. So we're on the march to the top. We're not at the top yet. So I think we can still become better and larger and can continue to make an impact. And this is why we believe we've used the IHI funding well in order to build this ecosystem, to create the stakeholder community, and now we're ready to also to deliver a continuation and really immerse even more into this phase of dissemination and exploitation in order to then also give back in terms of new entities, new jobs, new clinical trials, and also valorization and revenue for Europe in this exciting field of cell and gene therapy. Let's go 1 slide forward. This is some of the formats that we have tested in the project period and that we find particularly useful. Of course, roundtable discussions, also at meetings, also for visibility. The Science Cafe as a multi-stakeholder, multi-expert forum to discuss a specific topic and try to create a consensus and a path forward. Something that we call technology scan events, where we either focus on a particular technology topic or also give a particular entity the chance to present a new technology to multiple stakeholders in the field in order to increase its adoption, maybe also refinement. And of course, joint workshops with different experts, either scientific and medical versus patient, public and regulatory, or between different disease areas. Let's go 1 forward. This is increasingly important because, as I mentioned earlier, this field of CAR-T and also TCR-T therapy is not anymore limited to hematology and oncology, but it's also taking a bite on treating autoimmune diseases, for example, also infectious diseases, even cardiovascular or neurological diseases are in scope. And there are additional patient groups and stakeholder communities that we are going to engage. And this is why we believe that there is still another prime time for T2Evolve ahead, and where we're trying to really expand the scope and also the impact that we can make to another level. So please, we're very grateful for the support from Osama and the team at the IMI and now IHI office that we've received. I think we've been able to work quite productively and be able to use the trust and also the funding wisely. And we're all highly motivated to now continue the work in the T2Evolve Association and also give more back also to the community in Europe over the next couple of years. We're quite active also in interacting with the other European consortia, both in the IHI initiative with IMSA, for example, but also in the Horizon Europe program, Certainty, and 8Path. Also, the aspect of manufacturing, and Sonja with EASYGEN is presenting the next project that I think is also leading the way for this field in Europe. And we're very grateful and full supporters of these EU funding schemes because they really make a difference, and they're also quite distinct because our colleagues in the US and Asia, they don't have these network grants and they envy us for this. So with this, I do thank you for your attention, and we'll be happy to take questions that you have.
Thank you very much, Michael. Very good presentation. And I mean, this is also a perfect example on how IHI and IMI projects are key to assemble these cross-sectorial and public-private partnerships in order to achieve this wide range of objectives. I mean, moving from improving patients' information to engaging in regulatory framework, developing new preclinical models, and so on. I mean, this is really impressive work that you did. Thank you very much. So it's the perfect transition that you've made now, speaking about the synergies between T2Evolve and other projects, and specifically EASYGEN, which will be the focus of the next presentation. So EASYGEN is a project that has just started a few months ago. So the presentation will be much more focused on the objective of the project rather than on the results, because obviously it just started a few months ago. To do so, we have the great pleasure to welcome Sonja Steppen from Fresenius. Sonja is the head of research office at Fresenius, and she is principal investigator and the project coordinator of the EASYGEN projects. Sonja, the floor is yours.
Thank you so much for the introduction. Thank you for supporting this project. I'm pleased to be here together with my co-presenters, and we will make sure that we build on everything that was already discussed and described today. I start with a slide, and some of this is now repetition, but I will go over this quickly to highlight the burden of cancer again, of liquid cancer. So leukemia is the 11th leading cause of cancer deaths across Europe. If we look into numbers, here on this slide we have 14 different cancer types, but the incidences, and we heard this already from Osama this morning, the incidence of leukemia is increasing. From 2022, it's projected to rise 21% until 2040 across Europe, and the same is unfortunately true for multiple leukemia, and I have this on the next slides. For multiple leukemia, it's even higher, it's almost 23%. This is data from Europe, 27 countries, both sexes, all ages. If we go to the next slide, we can directly jump into the topics that was discussed the whole morning. So the CAR-T revolution started in 2017 with the first therapy available, covering science, breakthrough therapy, immunotherapies. This therapy is personalized, but this leads us already to the problem of the therapy, because each and every batch is produced individually, and automation and batch production is not possible. That's why we think the process must change. And many have already discussed this in publications. On the next slides, I will give you a short introduction into the process, and I will also go into more detail during my presentation. So the current process is done centralized. You have apheresis, then cells are activated, transduced with a viral. It takes up to 6 weeks. Cells have to be expanded, and then cells are re-infused into the patient. This could take up to 6 weeks, and this time is way too long. If we go into the next slide, I can give you more details, more numbers. So we tried to put all products on a slide that are already out there. There are 7 approved CAR-T therapies out there as of September 2025. Those products are available for different types of liquid cancer, but it shows also that 7 products is not enough. 7 approval does not guarantee access. So access is a huge problem. We heard this from Michael Hudecek, and I will come on add also numbers on this topic. So, but first of all, let me also point out that CAR-T therapy can deliver up to 3 times better responses than standard of care. And this innovation is outstanding, but it does not even this great. One should think that there should be an adoption very, very easily, but even though a 3 times better response does not guarantee access. Access is, however, and this is described on the next slide, limited to a couple of patients only. Globally, there are numbers out there that there are around 42 patients treated. So this slide breaks it down into each and every therapy. You can see that in 2017, there were only 32 patients treated. The numbers are increasing with around 10,000 in 2024 and most likely 12,000 in 2025. But 42,000 treated patients worldwide do not help all the million patients waiting for this revolutionary treatment. So let's look a little bit more deeper into the situation in Europe. So in Europe, the numbers are even more concerning. It's only 18% of eligible European patients who receive CAR-T therapy. The countries are very different, so the numbers are not the same. You can see that France is leading, Italy is falling behind, and we are trying to understand at the moment the reasons behind those differences in those countries and are doing stakeholder interviews to figuring out the different processes, the different delays maybe in process, the different healthcare system. On the next slide, we wanted to give you an overview. What are the prices for those therapies? Nobody has touched this yet. So in Germany, the prices range for Kymriah, and I showed this was the first product on the market by Novartis, from 266K, and then this goes up to 351K for Abecma. Highlighting that the price is extremely out of range. What are the reasons behind those high prices? As I said earlier, the manufacturing process is complicated. There is no economy of scale possible. Most of those products are produced by a virus, by lentivirus. 2 of them are produced with another virus. Centralized CAR-T manufacturing and complex logistics add up to this very high price. And this is only the example for Germany. If you look into the US, the prices for CAR-T therapy are even higher. If we come to the next slide, I would like to add more numbers to this dilemma. And I'm now doing a little bit of repetition, but I think it's all about CAR-T therapy, and this should be allowed today. So in Europe, there are 14,900 patients logged in the EBMT registry as of August 2025. So this is smaller than 20% of those eligible patients out there. We had this on the slide before. The time for production takes up to 6 weeks. And this is a big problem for patients that are proceeding during the manufacturing time. And 25% of those high-risk patients are knocked off treatment. Yeah, so making the treatment, the manufacturing time shorter would allow to also give access to those patients. Michael also highlighted this on one of his slides. So 30% of total cost of care is logistics, is delays, is bridging therapy. That's why decentralizing and bringing this therapy to the hospital might be very worthwhile to reduce costs and make this therapy more accessible. On the next slide, again, we have some more numbers for you. Michael showed the differences between Europe, China, and the US. We came up with 1,450 CAR-T assets out there, most likely tested in multiple trials. But most of those trials at the moment are done with lentiviral material, with viral material. And this pipeline growth will soon outstrip the viral plant capacities. And there is an urgent need for innovation to change this. So the IHI Call 7 focused on reducing workload in hospitals, and we believe we will achieve this because we would like to bring the process through the hospital, make the process easier, reduce the workforce load, include automation, and improve patient reach. And therefore, we are grateful for the IHI support and believe that the EASYGEN will tick all 3 boxes and make sure we can deliver on our vision. On the next slide, I promised I will go a little bit more into the detail of CAR-T today. So for those of you who are not in the topic, I know this is a busy slide. So we tried to nail it down on the 10 major points, the 10 major steps that are involved in CAR-T manufacturing today. Again, the process takes up to 6 weeks. Multiple steps, washing cells, T-cell activation, genetic modification, expansion, quality control, precision reinfusion into the patient. So this is a busy process, and it's done centralized away from the patient, not in the hospital yet. There are examples in Spain, we are deeply aware, but the majority of products is produced centralized, and that's why it takes so long to get those products to the patient. On the next slide, we show and imagine this as a dashboard, as a whiteboard, how we scribbled the process, how we believe we can build up a new process, making sure that this long process is cut down, is combined, is stripped, is shortened. And we would like to bring the process to the hospital, decentralized, virus-free, designed for usage in hospital, easy to use. And our ambition is to make this possible in smaller than 24 hours. On the next slide, that's an ambitious, bold vision that we have. And therefore, we brought together a cross-functional force to tackle those challenges. We want to make sure we can talk to very different disciplines. So we have 18 partners across 8 countries to enable this. We have process experts, scientists, of course, clinicians, hematologists, but also regulators, biotech experts, manufacturing experts, automation experts. And we are a strong believer in synergies. And with those different disciplines, we strongly believe that we will be able to bring the field forward and make an impact. And on the next slide, I show you our 10 working packages, or how we call it, the EASYGEN powerhouse. So we have to make sure that this is coordinated, and Fresenius is very proud to act as a project office here to channel all those different communities, make sure we can amplify the results, make sure we can build on each other's activity. And this is how it is split down. As I already said, we have process management, we have health economies, we are currently doing stakeholder interviews to completely understand why the process is sometimes delayed. And we will pick up everything that we can find and where we can build upon to channel this into our activities and make sure patient access will be available in the future and will change. At the end of our journey in the last year, we have with our Helios hospital chain then a real evaluation phase. I make clear we do not do a clinical trial, but we would like to do mock-ups tested in the hospital of Helios together with our partners and make sure we validate the whole process so it could be picked up for clinical trials after this completion day of this consortium. On the next slide, I would like to highlight the potential impact of the EASYGEN project. So we would like to make sure that cancer immunotherapy is cost effective, and we would like to achieve together with our partners a democratization. We would like to reduce costs by going non-viral. We would like to reduce manufacturing time and thus enabling increased patient access. And with this vision and with this impact slide, I also would like to come back to the Emily Whitehead story that Michael already picked up nicely. And on the next slide, you see again Emily Whitehead, so first child worldwide that was treated with CAR-T therapy. So her story moved all of us very deeply and inspired a complete industry, I would say. So Emily Whitehead was diagnosed 2010. She was she was going into relapse. Her parents had no other chance than subscribing her to a clinical trial. She was treated with Kymriah. So at this point, it was not Kymriah yet, what later became Kymriah. And she was in good shape after this treatment. And that's even more powerful 10 years later. She's cancer-free. She's 25 years today. She's a strong advocate for patient access. She was together with her family. She founded the Emily Whitehead Foundation. Patient advocacy and patient access are the most important to her, and we are very glad that she is also looking at EASYGEN and collaborating with us and making sure we are doing the right job here. This leads me to the next slide, and there's nothing more to say at this short presentation with a project that just started but has a very bold ambition. So acknowledgement goes to the IHI office for supporting our visions, for supporting this project. The team couldn't be motivated more. You have all partners on there. And with this said, I'm happy to take any questions. Thank you.
Thank you very much, Sonia, for this very great presentations. And you showed how EASYGEN and is ambitious and the really ambitious goals that you've set. And I think only a public-private partnership IHI project could achieve this kind of ambitious goals at this scale and significance. It's really great project that you presented today. As you can see, so we have presented today 3 different projects. And as I said during the introduction, we have many other projects working on different indications in cancer, please scan the QR code which is on this slide if you want to learn more about other IHI and IMI projects working in oncology. And also, I draw your attention to a past event, a past webinar, where we have presented other projects working on other indication and fields in oncology, such as prostate cancer, but also interventional oncology and liquid biopsy. So now is the time for Q&A. So we have received a number of questions that I'm going to read to our distinguished speakers. So I think this one is for Harmony from Ellen. So I assume, but grateful for confirmation, that you work with some or you are based in hospital units which are members of the European reference networks, EuroBloodNet. Lars, Jesus?
Lars, do you want to reply? Or...
Yeah, I mean, I know that several of the members of Harmony are also members of the EuroBloodNet initiative, like the Charité, for example, is. Yeah. But I could not detail really how many out of the 100. I'm not sure.
I will add that some, even the coordinators of EuroBloodNet were also members of in that case, of the MDS pillar. So in fact, we were mainly focused on malignancies, on blood cancers, while EuroBloodNet is mainly covering any kind of all the other hematological diseases, covering red cell diseases and some of the benign white cell as aplasia and some others.
Thank you. Another question from Sven. It's for, everyone, but I think it's more relevant for you, Michael, for T2Evolve, because you touched upon the cross transversality of the work of T2Evolve. It says, an open question to anyone, really. Today's webinar is about blood cancers, which is a very important set of indications. To what extent are the blood cancer a unique indication set, and to what extent can ideas and approaches be generalized across other cancers too?
Yeah, I think it's an important question. I mean, often blood cancers, are kind of paradigmatic because it's relatively easy to measure response, right? You can take a blood sample and see if there is a leukemia regression or not. Expanding on this thinking, I think many of the models that we've developed and the concepts can also be extrapolated to, in our case, immune cell therapy in oncology. And then also with some adjustments also in the non-malignant disease areas. For example, and we have an interest also in treating certain infections, be that pathogens like Aspergillus with immune cell therapies. And there's a similar need then for models, for example, of the human lung to determine efficacy, but also potential toxicity that such immune therapy may exert. And we are currently using actually the same model that we're using for CAR T-cell therapy to treat lung cancer now to actually treat this pulmonary infection with Aspergillus. So I think there is a certain level of proximity and the ability to extrapolate into these other disease areas, even though some adjustments will have to be made in order to then reflect the disease. And then also to inform the clinical translation, because in other disease, fields of medicine, let's say the benefit and risk assessment, right, especially with novel innovative therapies, may also be different compared to hematology and oncology, where typically also the risk that we're willing to take with an experimental therapy is somewhat higher because of the severe prognosis that patients have. I mean, that also goes to Sonia, right, with manufacturing. Of course, we have a strong interest in manufacturing immune cell therapies for hem-onc, but also now with these insights have the ability to really roll this treatment out to really other fields of medicine where there is also probably even more patients. Sonia?
I can only agree. So the manufacturing topic is certainly in the center of everything, and I know many many companies are working in this field, trying to bring this innovation forward. The need could not be higher. We are committed here, but it's an ambitious goal. We are deeply aware.
Thank you very much. Sonia, while you're speaking, there is a question for you for EASYGEN. So you showed in a map that patients' access in some European countries are much higher, for instance, in France, with 30% of eligible patients, and that in other countries, such as Italy or UK and Germany, it's not the same percentage. They're quite behind. And you mentioned that you're looking into the reasons for this, but do you have any assumptions on what could explain such a disparity?
Different healthcare system, different number of treatment centers. So in Germany, I think we have 42 treatment centers. All of this, so access in terms of how many kilometers people have or patients have to go to a hospital, how accessible is a hospital, how many hospitals are offering CAR-T therapy. So the numbers I presented were from a recent study or a recent article from IQVIA Health Institution. We also reached out to them to collaborate for more details. And yeah, we are trying to understand those numbers. More than I just said, we don't know yet, but we are eager to harmonize and to figure out, okay, what can we learn from France, for example? If they have a higher number, they must do something better.
Thank you, Sonia. A question for Harmony. I think the tools for data integration and research data management developed for Harmony immensely useful for other domains. Are those models or lessons learned from them published anywhere?
So I want just to reply this. So it is possible to use this system from the web, and this is a totally open tool. So from Harmony, also I want to stress here that I think the IHI projects are really overarching projects, trying to translate the knowledge from one disease and to expand this to be used in another diseases. In that way, the Harmony platform is providing a tool for all the people, for all clinicians, for all patients that are interested to follow this type of data lake with well-structured data, well-curated data, in order to have these tools and applying these tools as, for instance, a visualization tool that Lars already explained to all of us, and also the prediction tools. Some of these prediction tools are also based on artificial intelligence methodologies, different from the AML tool, from the myeloma tool, because we have to use a different tools in order to take the best accuracy from the data that we already collected. This platform can be used for another disease, cancer disease or other disease, in order to collect as much data as possible in order to exploit this data. And the risk calculation tools that are based on artificial intelligence methodologies, Bayesian methodologies, deep learning methodologies, artificial network. So these are available tools that could be used in any place in the world.
Perhaps I can add, but
maybe.
But I also understood the question perhaps also in the way on with regard to how we solved the data anonymization and the harmonization process. I think all these things, of course, were put together in the reports which we sent to the European Union. But I think some of the things might still be worth also for being published as a white paper, for example, and we could think about doing that. That might be very helpful to the research community, like lessons learned from Harmony, and make some of the, let's say, workflows which we have established, how you deal with data harmonization, more accessible. Perhaps this is, I think that's a good idea that we could still publish some of the things which we have developed, but that are not yet out there. Yeah.
It'd be great to, yeah, of course, to publish some lessons learned on the barriers that you've kind of crossed in the past, and how you overcame these barriers. Then another question from Sven as well. For T2Evolve, he said, I'm interested in standardization. Where are these soaps available? And is there any formal standardization work being performed?
Yeah, so some of the standards that we've created are published, or there's these papers reporting on how these standards were developed. And if you reach out to the T2Evolve product office, we'll be able to share those things that are completed already. And there's more things in the making. So please reach out to us, and we'll be happy to share what is available. Yeah.
Thank you, Michael. Another question from Peter for T2Evolve, again, while you're at it, Michael. Mm-hmm. So I think it's more of a comment and then kind of a question. Isn't there a lot of double work in the CAR-T trials? There are around 700 trials worldwide. How many CAR-T applications are there?
Yeah, I mean, that's a thing that I can maybe comment on, but you know, there's certainly a lot of activity. Maybe there's also some trials that may seem redundant, but there's also, you know, of course, a desire and motivation to bring CAR-T therapy to different geographical regions, right? And if there's an IND active at one cancer center in the world and another cancer center and patients on the other side of the world also have the desire to benefit from this treatment, often it is necessary to launch another clinical trial in that geographical region in order to work towards an approval or a hospital exemption or some format where patients then get access. So it's also part of the effort of trying to give patients as many sites as possible access to this treatment. This is why we see some things that may seem redundant. Mm.
Thank you, Michael. Maybe something that you and Sonia could address. T2Evolve and EASYGEN projects all working in the same field, in gene T cells. How, because you mentioned, Michael, the synergies that are ongoing with other projects, how do you plan any collaboration or with EASYGEN, for instance?
Yeah,
she's just starting.
Yeah, I can maybe start, and then Sonia can also follow on. I mean, we've now been also considering the topic of GMP manufacturing, in-process controls and release controls, and some harmonization there, including also the particular assays that are used, for example, to determine sterility, potency, identity of the CAR product. And these are things that can essentially be taken forward and reassessed and adjusted to EASYGEN. EASYGEN, of course, is then also pushing the manufacturing concept itself, where the manufacturing will be particularly fast and also directly at the patient bedside, extracorporeal. So there will some adaptation be necessary. But this is one aspect. And I think we've created the stakeholder community. There's more than 100 entities, and I hear from Sonia that there's also, you know, such an interest in EASYGEN now that we should maybe look at what are the partners and what is an efficient format to engage all of these partners. First, to serve their interests and to support them, but also, you know, still maintain a consortium that is, you know, agile and ready to also execute. And interested to hear what Sonia has to say. Mm-hmm.
So there is huge interest ever since we released the press release 4 weeks ago. Our LinkedIn profiles are overfloated with emails. Academia wants to join, industry wants to join, patients are giving us insights. People are applying through different channels to be part of this initiative. So we are trying to talk to all of them, trying to figure out how can we collaborate. But as Michael said, at the end, if we have 100 partners, this is not manageable anymore. But for sure, we will be also talking to manufacturers, companies, and academia working in the autoimmune disease field. There are already platforms that enable a shorter manufacturing process. And as I said, we are a strong believer of synergies, of collaboration, and not a single company, not a single institute will be able to solve all the problems out there. And we are trying to incorporate as much as the knowledge that is already on the table into what EASYGEN would like to achieve. And this goes beyond liquid cancer for sure.
Thank you, Sonia. One last question for Harmony. You explained the possibility for researchers to submit research questions to the Harmony platform. Can you please provide more information on the modalities such as access fees and the timelines to get feedback?
We are open to receive questions. Any kind of question will be of great value for us, and usually the time to respond to the question and to ask for is around 4 weeks. And you have the possibility to take access to the Harmony Alliance Foundation by clicking on the web page, and this is the touch point, and the contact is already written there. There is an office who is receiving the questions, and we are open to receive questions, to receive data sets, and to share and to take to go in this idea of data sharing.
And for academic partners, it's free. And if you are an industry partner, of course, depending on what type of data you want to use from Harmony, yeah, we would ask for some support for the platform. Yeah.
Thank you. I guess all this information is available on the website, or if they reach out to you. Okay.
Yeah.
Thank you. Maybe one last thing. For Harmony, you have collected an impressive amount of data. Do you plan to continue collecting more data? And do you also collect follow-up data to complement the existing data sets from existing patients in your platform?
Yeah, the general idea is to continue in the way. So, and now the new ideas coming for the foundation is to collect more data with new therapies. That this is really something that it is really very important to have this access to these new drugs coming to the different hematological diseases. So we are open to receive data, and for any kind of data coming from trials, from real world, from your hospitals, they are more than welcome. Please don't hesitate in contact us and approach us. The project is alive, the project is growing, and the idea is continue with the project, with the platform, and with the collaboration between the partners.
Thank you. So we are reaching the end of this webinar. I would like to thank the speakers really for your time and for the preparation of these presentations. It's really great that you could make it. And I would also like to thank the audience for attending this webinar, and also a special thanks to my colleagues from the communication team, Nassima and Catherine, who have been instrumental in organizing and running this event. Thank you all very much.